Hello and welcome back. Today we'll be covering lipids. The structures of lipids is fairly variable, but in general there's a few rules that they all follow. So they can be saturated or unsaturated, and to further classify them, the essential lipids are the ones that we cannot synthesize within our bodies, and they are polysaturated with two or more double bonds. And a few examples are the linoleic acid and the alpha linoleic acid. The non-essential, the non-essential lipids are the lipids that we can synthesize within the body, and those are generally saturated or monosaturated fats, and they are arachidic acid, stearic acid, and palmitic acid. And the most naturally occurring unsaturated fatty acids have the cis double bonds. And that's particularly important for us because we as humans, we can metabolize the cis double bonds. So these are the important structures. Laurate is what we use to make soap, and acetate is a precursor to the even number of fatty acids, while plants make the odd number fatty acids. Odd number fatty acids are non-essential, but we cannot make them, and neither can we metabolize them, right? So humans can't degrade trans fatty acids, and this is the problematic lipids and fats that everybody tells us to avoid because they accumulate in the body, causing the obesity and some metabolic disorders because we cannot get rid of the trans fatty acids. And the neutral fats store metabolic fuels. So what are neutral fats? They stay in the subcutaneous tissues. They are an ester of fatty acids with a glycerol attached. And its main role is a structural role. For example, it keeps the kidneys in place. If you are very anorexic, you don't have enough subcutaneous tissue, the kidneys will descend in a syndrome called renotosis. As you can see here, these are the renal artery, and the kidneys have descended all the way into the pelvic cavity. They also play a role in thermal and protection. So the neutral fats are classified by their total number of fatty acids into monoacylglycerol, diacylglycerol, and triacylglycerols. So alpha monoacylglycerol is the main product of digestion. Triacylglycerol is the most abundant fat in nature. This is the one that we consume the most of. Diacylglycerol is a second messenger precursor. This plays a role in cell signaling and hormone regulation. And palmitic acid is the only fatty acid that we humans can make de novo. Phosphoglycerides are esters of diacylglycerols with a phosphate and a alcohol attached. They play a large role in cell membranes. Here are a few examples: lecithin, phosphatidylenolamine, phosphatidylserine, phosphatidylglycerol, inositol, and cardiolipin and plasmalogin. These are all under the phosphoglycerides, and they all contained these two elements. Okay, so phosphatidyl is the precursor of all the phospholipids. So you can see here, these are the functional groups attached to the phosphate in the phosphodiester linkage. And the phosphatidylserine and ethylenamine are precursors to acetylcholine, which will make the phosphatidylcholine. And as you can see, they're all kind of linked together with the phosphodiester linkage. Here. Plasmalogin is a fatty aldehyde, and it has an ether linkage. It's found in the cell membrane, in the nervous, immune, and cardiovascular system. It also plays a role in clotting. So these are the functions of various phospholipids. They are lecithin is the main component of surfactant. They are starting to be made by week 28 in human development. Phosphatidylglycerol is part of the cell membrane, but it also is a component of surfactant. Phosphatidyl and inositol is a secondary messenger. This is one of them. A cardiolipin is a mitochondrial membrane component, and it plays a role in blood clotting. And a decrease in the cardiolipin levels will cause mitochondrial dysfunction during aging or heart failure or hyperthyroidism and Barth syndrome. And then plasmalogen is in the brain and nerves, and they also play a role in blood clotting. Okay, so sphingolipids they are precursors or components of sphingomyelin, sulfolipids, cerebrosides, and gangliosides. Generally, it's a lipid of a 18 carbon chain and a alcohol attached to it, using like a carbamide linkage. And ceramides are the intermediates in sphingolipid synthesis. And sphingomyelin has a choline in it, and it's polar, similar to the phospholipid. And some of the sphingomyelin are in the cell membrane, but generally they are in the myelin sheath. Galactosylceramide is a major glucosphingolipid in the brain, while the glucosylceramide is in all the other tissues. Lysosomal storage disease occurs because of the lack of an enzyme. There's a bunch of them. Remember in the other lectures we went through all of the individual types, but in general the body can't hydrolyze the glycosidic linkage in the cerebrosides. Gaucher's disease is a disease where the normal bone tissue is replaced with beta-glucocerebroside, making the bones fatty and very weak. So they are prone to breakage and stunted growth and deformation. Globosides have ceramide and oligosaccharides in it. Sphingomyelin, once again, are in the nerves, particularly the Schwann's sheath. Gangliosides are made of ceramide along with oligosaccharides and sialic acid. Cerebrosides are in the brain, particularly the white matter, and the cerebral cortex is where they're really concentrated. Sulfolipids are in the cell membranes and the neuromembranes of the axons. Globosides are also in the neuromembrane, generally in the cell body. Gangliosides are in the gray matter of the brain and the neurobodies. They are concentrated in the ganglia. Gangliosides and globosides play a role in antigen and cell recognition. Isoprenoids are derivatives of isoprenes, and their main role are scents, because they make the terpenes, right? Let's see from here, like geraniol comes from the geranium flower, so they have a certain scent. I don't know if you've ever smelled it before, but to me it smells a little bit like dirt. So this is the scent that our brains will pick up. Linalool is like a citrusy scent. I'm pretty sure it comes from some form of citrus fruit. Citronella, of course, comes from the citronella plant. And if anybody has lived in anywhere that has a lot of bugs during the summer, like Florida, everybody burns a citronella candle. So the biologic terpenes depend on the functional groups. of course, and the biological terpenes, like especially in the human bodies, these are the examples: the coenzyme Q and vitamin A, K, and E. Okay, so steroids are very, very important. They are the derivatives of the steroid rings. They're all made from cholesterol. This is the general structure. Of course, they have an alcohol attached, generally at carbon atom number 17, and there's a double bond. It can be either from 4 to 5 or 5 to 6, and so of course sometimes they have a side chain, and they have a generally like about like 18 through almost like 30 chain length. So sterols, they're the carbon atoms can be like 27 to 30, and then the chain will be 8 to 13. Bile acids have 24 carbons, and those have the acid groups. Progesterone, mineralocorticoids, and glucocorticoids have 21 carbon atoms. Androgens have 19, estrogen has 18, and cholesterol has 27. And this is the general structure of cholesterol right here. So cholesterol is not the great evil that we have been led to believe. It is not really to blame for atherosclerosis. It has a huge role in digestion and signaling and hormones. Bile salts are made out of cholesterol, and those have a taurine attached instead of glycine. And without the bile salts, we wouldn't be able to digest lipids at all. The diarrhea will occur because cholesterol from the lipids from our diet cannot be absorbed. Progesterone has a ketone. Cortisol, aldosterone, all of those are made from progesterone. And since progesterone is that kind of precursor, it has some mineralocorticoid and glucocorticoid effects. So in women, of course, we have higher levels of progesterone, particularly during pregnancy. And for that, some people can have gestational or transient diabetes and hypertension because of the effects of the increased amount of progesterone. And like I said before, progesterone is the precursor to aldosterone, so that can increase the blood pressure. And cholesterol controls, of course, like the blood pressure, gestation itself, because you need progesterone to maintain the endometrium for the fetus to survive, and it also controls the sugar level. Okay, so waxes are the true evil of the biological molecules, because these are the ones that do lead to atherosclerosis. They are made out of ester of a fatty acid along with the alcohol and a glycerol. It's created in the skin for lubrication and protection, and the part of the wax that is bad is the cholesterol ester, not cholesterol itself, just cholesterol ester. So it causes the atherosclerosis because the body isn't able to properly get rid of it or use the amount of cholesterol ingested. So since the body can't get rid of it, it's not able to it's not able to regulate the amount because we eat more every day, right? And it just accumulates because the body isn't able to process the cholesterol ester. Eicosanoids are derivatives of arachidonates or arachidonic acid, and those can make prostaglandins, which are the local hormones, and then or it can make the trenes, which play a role in the immune system. And then, as you can see, ligotrienes and glutathione, as you can see, immune system. So make the oxides in order to break down foreign materials, old cells, or bacterial invaders. From prostaglandin, it also makes thromboxans, which play a role in blood clotting, and it can also make prostacyclin, which is a local hormone as well. So the local hormones will act within a nearby region where it is first secreted. And that's it for now. Thank you for your attention and good luck studying.